Fourth, the regulation of enzyme release from the pancreas. Coming out of the stomach, if it's a fat or protein-rich meal, will be amino acids and fatty acids. Either one will bind to secretory cells in the intestinal wall. That triggers the release of cholecystokinin, CCK. Cholecystokinin has two different effects. It triggers release of enzymes, secretion of enzymes, held in granules in enzyme-secreting cells and then released as inactive zymogens, activated in the intestine. A range of enzymes including proteases and lipases and many others. Cholecystokinin also causes bile secretion, bile from the liver stored in the gallbladder, causes smooth muscle contractions that move bile out. The enzymes break down proteins. If there are amino acids present, then there are presumably also proteins, and the enzymes break down those proteins as well as fatty acids. The bile acts on droplets of fatty acids. Droplets of fats form because they don't mix well with water. A lipase can only attack the surface molecules in a droplet of fat, and a droplet of fat can be quite large. The bile, by having a nonpolar and a polar end, salts of cholesterol, the cholesterol molecule with a charged group on it. The salts of cholesterol with their nonpolar end are on the outside of the fat. Their polar end interacts with water. So the salts of cholesterol coat the droplet. They start crowding in to get closer to the fat because these nonpolar ends do not interact well with water. That causes the fat to start stretching. And eventually it breaks into two smaller droplets, each of which is coated. That process continues. So with excess salts of cholesterol, they keep coating, they keep crowding next to each other, and they increase the surface area of the fat, eventually cause it to split into two smaller droplets. But what begin as fairly large droplets become over time, because of the bile, smaller and smaller fat droplets, which means there's a much higher surface area for lipases to attack. If the fats are still present in high abundance later down in the intestine, additional secretory cells respond to those fatty acids by releasing another hormone, gastrointestinal peptide, GIP, in response to fats. Fats that are still present further down the intestine cause GIP release. GIP acts to slow down the whole process. GIP acts to inhibit movements, less movement, also inhibits hydrochloric acid release, and finally inhibits pepsinogen release. 3A inhibits movement, 3B inhibits hydrochloric acid production, and 3C inhibits pepsinogen. That slows down movements of material out of the stomach, and with less material leaving the stomach entering the intestine, that slows down movement through the intestine. That slower movement provides more time for the bile salts and the lipases to break down fats. So a response to a very high fat diet slows down movement of material through the intestine, giving much more time to absorb all of those fats.